Hello dear students, welcome to the class. Are you ready for 10 more MCQs for molecular basis of inheritance? Let's begin. Right? First question you can see on the screen. Okay, they are asking that during DNA and RNA synthesis, okay, a nucleotide is always attached to the newly formed strand at which end, right? So they are asking here that will you add that at 5' end of new strand, or 3' end of new strand, or 5' end of coding strand, or 3' end of template strand. Do you know the answer? I think in last class also I have explained this, something related to this, right? So if you remember one thing, okay, if you remember one thing, you can never do mistake in this question. First thing you have to note down is all polynucleotides, okay, all polynucleotides. And if I am writing all the polynucleotides, what do I mean? I mean both DNA as well as RNA. Okay, whether it is DNA or RNA, will all the polynucleotides will be synthesized. Okay, all the polynucleotides will be synthesized in only 5' to 3' direction. Okay, this is what you have to learn. Okay, 5' to 3' direction. Second point you have to remember, 3' OH. Okay, 3' OH is required. 3' OH is required for chain elongation of polynucleotide. Okay, 3' OH is needed for the chain elongation of polynucleotide. So if you are making DNA or RNA, you require 3' OH. That's why you can synthesize it from 5' to 3' direction only, right? So let's talk about first DNA, right? If I talk about DNA synthesis, which is the first type of polynucleotide, right? In DNA synthesis, both 5' to 3' strand, right? Both the strands which are there, they will be acting like a parental strand. Okay, let's suppose this is my 3' to 5' template strand, this is 5' to 3' template strand. Both will act like parents, right? And on this parental strand, I will be synthesizing my new DNA in this direction, 5' to 3' in both the strands. So it means that my new nucleotide will come and attach on this side, right? On this side, right? That's why we require 3' OH always, because next nucleotide will always come and attach from the 3'. From nucleotide's 5' position, it will interact with already existing 3' OH of the existing nucleotide. Both will join and they will make 3' 5' phosphodiester bond. Okay, understood? So for this, you require, this is your new strand, right? This is your new strand, right? So you require 3' OH in new strand, right? Fine. Let's talk about, okay, now this is done. Now let's talk about RNA synthesis. This is in DNA synthesis. In RNA synthesis, what is the case? In RNA synthesis, any one of these strands will act like a template, right? Not both. Let's suppose this is 5' to 3' strand and this is the opposite one. But synthesis direction is shown by the arrow. This has been formed in this direction, nothing else, right? You tell me which will be copied from this, both or only one? I asked this question last turn also, but nobody answered it, right? So this one is acting like a template strand. This will act like template strand. Okay, using this as template, you will be making messenger RNA. Okay, and you will be making messenger RNA from 5' to 3' direction. So what you are able to see here, you are able to see that for making any kind of polynucleotide, you always require 3' end in the new strand. This is the new strand, right? You will not add anything on template strand. Can you? No, right? So what is the answer here? 3' end of new strand, right? Can you add anything to template strand? No. So you cannot choose this as an answer. Understood? Clear? Okay, let's move on to the second question. Second question is asking that gene regulation in eukaryotes may occur at all the given except which level, right? First, you tell me what is the meaning of gene expression? What is the meaning of gene expression? Gene expression means protein synthesis, right? Information which is present in the gene will become functional when you will be able to make protein from that information, right? And what is the meaning of protein synthesis? It simply means that a gene is showing its expression, right? A gene is showing its expression, what to do, right? That will be done by protein. Gene alone cannot do anything. Okay, but do you require protein same amount at all the time? No. So you need to regulate this, right? Okay, regulation of gene expression. Now, what is the meaning of regulation of gene expression? Means that let's suppose today uh, uh, okay, what do you mean by regulation of gene expression? Regulation of gene expression means that we do not require all the protein at all the times, right? Sometimes we require some proteins at much lower amount, sometimes we require that protein at higher amount, so their synthesis must be regulated, right? So that regulation exists in all the living organisms. Okay, every living organism has the you can say capacity or ability to regulate the expression of gene. Okay, it is there in prokaryotes also, and it is there in eukaryotes also. Which kind of regulation exists in prokaryotes? Only one. Okay, it is at transcription level. Okay, simply if you can increase or decrease the synthesis of your messenger RNA, you can increase or decrease the synthesis of protein also, right? But in eukaryote, it happens at 4 different levels. Okay, in eukaryote, it happens at 4 different levels. First level is transcription level. Second level, who will tell me? Modification level. Okay, modification of what? Modification level. Okay, because we make hnRNA, right? hnRNA in eukaryotes. We have split gene arrangement. We have introns, exons, everything. So we have 3 types of modifications: splicing, tailing, gapping. All 3 must be done. Then only the RNA will be ready to enter in the cytoplasm for translation. Next is transport of modified mRNA. Okay, transport of modified mRNA nucleus nucleus to cytoplasm. If this step also will be like naturally, normally all of this will happen, right? But if you want to reduce the synthesis of your protein, what we can do? We can do any mistake or any problem here, right? Our cell can do, we will not do actually. Our nucleus, inside that either something will happen at transcription level, so messenger RNA formation will be affected, as a result protein synthesis will be affected. hnRNA, okay, if you are not able to convert this heteronuclear RNA into mRNA, mature messenger RNA or modified messenger RNA, then also protein synthesis will not happen, right? Or you do not allow your modified mRNA to leave the nucleus. At that case, in that case also, protein synthesis will get affected, right? And then at last, translation level. What all things can happen at translation level? Let's suppose the ribosomes are not binding, okay, or amino acids are not there, or charging of amino acid on tRNA has not been done, right? tRNA are not charged with your respective amino acid. In any of these cases, what will happen? Translation will get affected, and as a result, protein synthesis will be reduced, right? So regulation is there, like increasing the protein synthesis or decreasing the protein synthesis rate, that is called regulation, right? So in all of these things, you can see it happens in both prokaryotes and eukaryotes, right? But in both the cases, do you have any regulation at replication level? No. How you can do regulation at replication level? No need, right? Replication just mean that from DNA you are making DNA, right? DNA alone can do anything in synthesis of protein? No, right? So that's why this step you do not require. Okay, whether it is eukaryote or prokaryote, this is not needed for regulation of any kind of gene expression. Understood. Only these 4 in eukaryotes and only 1 in prokaryote is needed. Okay, one more question you can see on the screen, which is very easy question. Sequencing of DNA of non-human organisms can help solving challenges in healthcare, energy production, bioremediation. Which option is correct from this? This you have to do. Okay, very, very easy. Okay, if you consider sequencing of DNA of non-human organisms. Okay, so you know that if we have done DNA sequencing, right? We have done DNA sequencing. First, to with the help of HGP, right? We have done sequencing in humans. Okay, so human DNA has been sequenced. Okay, then there are certain non-human model organisms. Okay, there are certain non-human model organisms. Can you name me one by one? Their sequencing of genome also has been done. First of all, bacteria. Which is that bacteria? Anyone remember? You only have studied in the first part of this chapter, E. coli has how many base pairs? It means you have sequenced it, right? Yeast sequencing has been done. Then one nematode is there, non-pathogenic nematode, C. elegans. Okay, its sequencing has been done. Then you all know about Drosophila. In the previous chapter, you have learned about it. Fruit fly, right? This also sequencing has been done, right? Along with that, you have to remember certain plants like rice, okay? And one more model plant is there, which is called Arabidopsis, okay? These are certain different types of non-human organisms, other organisms other than human, where the sequencing of the entire genome has been completed, right? And why we have done this? Because sequencing of these genome can help us understand more about them, right? And we can utilize such kind of information in industries like healthcare, for energy production, for bioremediation or environmental remediation, right? So all ABC is correct. And you can see this line directly given in NCERT here. Just see this. Okay, they have mentioned about DNA sequences can lead to understanding of the natural capabilities that can be applied towards solving challenges. Okay, solving challenges in healthcare, agriculture, energy production, and environmental remediation. The word only has been changed here. Instead of environmental remediation, they have mentioned bioremediation. Okay, what's the meaning of this term? Okay, I will help you in explaining with respect to environmental remediation. There are certain plants, there are certain bacteria, there are certain prokaryotes also, which actually take up the toxic, you can say, chemicals from the, you can say, soil, right? They will actually take it inside them, right? They will absorb it, or by any method, they will take it up and they will metabolize them, right? So this process is called bioremediation, right? If you are removing some toxic chemicals like arsenic and all, right? Some plants are there which will absorb them and eliminate them from the soil. So they, if we know that which genes are responsible for this, right, how they are metabolizing these toxic chemicals in their body, so these all informations are very helpful for us, right? If we know that, okay, this part of our DNA, or this part of the DNA of bacteria, or this part of DNA of any plant is able to do all this, and we uh, take that DNA and incorporate in some other crops and all, then we can remove the toxicity of certain soil. We can use that land for agriculture purposes and all, right? So this is one of the thing by which you can understand that why we have sequenced other organisms also, because we can have more information for healthcare industry, for energy production, and for bioremediation. All these three you have to remember along with the name. Next time they can give question on this. Okay, find the odd one out with the help, like with respect to non-human model organism. Any other name they will add here, right? So you have to learn that along with human, we also have sequenced the DNA of all of these, right? Then supercoiling of DNA in E. coli cell is maintained by, and the supercoiled DNA is called what? Very, very easy question, right? You have learned packaging in eukaryotes, which you all know it is done by histone protein. But if you think about the packaging, okay, packaging of DNA in prokaryotes, that also is there, right? You have seen that always you will draw the DNA in prokaryotes in one side of the cell. You will not spread it, scatter it everywhere. No. So some sort of packaging happen in prokaryote also, right? This packaging of DNA in prokaryote occurs with the help of positively charged proteins. Okay, positively charged. I'm writing here histone-like proteins. Positively charged histone-like proteins. They are not histone, but their character is like functional character is like histone. They are together called polyamines. Okay, what is the name? Polyamines. Name is not given in NCERT, so only little bit out of NCERT questions I am bringing for you. Okay, polyamines. Only one option you can see where polyamine is mentioned, right? Another thing I will write here, packaged DNA in the form of loops, in the form of loops in prokaryotes, or coiled DNA in the form of eukaryote is called, or coiled DNA in prokaryote, it is called prochromosome or genophore, or NCERT writes it as nucleoid. Okay, so genophore is mentioned here, no problem. Okay, second blank, everything is correct, but we have to find the answer from the first blank itself, right? Polyamine and nucleoid. Okay, understood? This you must remember. Okay, after this question is very easy. You can see on your screen. You have solved this in one of the previous classes also. Please see what is the wrong one in this. Okay, please check here. Rest everything is correct here. Only option number 1 is wrong. Why? Because first of all, you tell me you have learned about this, right? I told you there is a bacteriophage which is this one. What is the genetic material which it contains? It is having single-stranded DNA, right? And how many base pairs are there in that? 5, 3, 8, 6 bases or nucleotide. Can I write base pair? No, right? Because it is having single-stranded DNA. So problem is here. We cannot write base pair here and we cannot write double-stranded DNA. This is an odd one. Rest all is correct. Okay. This is 10 raised to 6. This is 10 raised to 9. Right? This is correct only. Right? Now this question. In which case the TM value of DNA is highest? Now you'll be asking what is TM value? It is very easy. In chemistry maybe you have learned. TM value means what? Melting point you can consider. Okay, in which case you will be having highest TM value? Always remember, TM value of DNA is directly proportional to GC content of the DNA. Okay, GC content of DNA. Okay, TM value of DNA is always equivalent, sorry, it's always directionally proportional to GC content. Means if you have high GC content, you will be having very high TM value. Why? Okay, because guanine and cytosine, they are connected by 3 hydrogen bonds, right? So if you have more GC in content in your DNA, if you have more GC content in your DNA, what is going to happen? It will be having more hydrogen bonds, and hydrogen bonds are very strong bond. You have to you can say make the temperature very high, more heat you have to provide, then only you can break the bonds. So in this all 4 options, so which is that option where you can see cytosine guanine must be highest? You can see option number 4, right? It's if the DNA is having 40% cytosine, it will be having 40% guanine also, right? So it means 80% of the DNA is having GC content. Very high temperature will be required for you can say breaking the hydrogen bonds and making single stranded DNA, right? That's why option number D is the answer. Okay, now this question, just see this question, so you should answer, because in last class also I have discussed something related with this question. Okay, you tell me about purines and pyrimidines. Okay, so tell me in purines how many are there? Adenine and guanine. And in pyrimidines how many are there? Cytosine, thymine and uracil, right? Both are heterocyclic structures. Okay, both will be having ring structures, right? Both are heterocyclic structures, both are ringed structures. Both rings are made up of nitrogen and carbon. That's why heterocyclic they are, right? This you have to remember, this is single ringed structure. Oh, sorry. Purines are double ring structure. Okay, this you have to remember, they are double ringed structures. Okay, they have double ring structure. This is having single ring structure. Single ringed structure. Okay, if they are having double ringed structure, right, this is having 9 members in those two rings. 9 membered. Okay, and this one only 6 member. Okay, so this is 9 membered rings, and here 6 membered ring. So how adenine is different from cytosine? Adenine is different from cytosine because adenine is 9 membered ring. Rest all things are fine, right? 6 membered we cannot choose, because if cytosine they are writing first, then we can choose 6, right? See this diagram. Okay, this diagram you have seen earlier also, where I told you adenine and cytosine always interact with their 9 position to the first carbon of the sugar, but cytosine, uracil and thymine, they always interact with their 6th carbon. Okay, this I have discussed. Sorry, not 6th. They will interact with their first nitrogen. Okay, so that we have learned. Anyways, see the ring, right? 2 rings can you see here in the adenine and guanine? Can you see 2 rings? Okay, 1, 2. Here also 1, 2, but here only 1 membered ring. Okay, and 1 ring structure, sorry, and 6 carbons here, but here 9. You can see the numbering, right? Okay, understood. Now this translation process is always, okay, translation process is always dictated by RNA, represented by RNA, dictated by DNA, represented by DNA, dictated by DNA, represented by RNA, and dictated by RNA, represented by DNA. Okay, what is the meaning of this? It's very easy question. Its answer is in Francis Crick's, you can say, central dogma of life, that day we discussed, right? So you know that DNA is something which can only have the information to dictate to RNA, right? And RNA is something which is going to actually make protein, right? So information came to RNA from DNA, right? Information of DNA to make protein is represented by RNA, right? So what is the answer? Dictated by DNA but represented by RNA. Understood? Okay, information to make the protein is represented by RNA, but RNA is getting that information from where? When the DNA is dictating that information, right? Information from DNA is copied into RNA. Then this RNA will be making protein. Okay, fine. Now, this question, just see. Select the incorrect statement with respect to DNA replication. If you have learned the chapter very well, you can do this question, right? It's very, very easy. Okay, see. Let's suppose I am drawing here 2 strands of DNA which are ready to do DNA, you can say, replication. Okay, replication fork I have made. You tell me this particular strand, what you will call this strand? You will say this is a template strand, right, whose polarity is 3 prime to 5 prime. From open side always you will write. Okay, this one this is also a template strand. Please tell me the polarity of this strand. From opened side, from replication fork side, you will write 5 prime to 3 prime. Okay, on this strand, when you have to make DNA, can you easily make the DNA from this to this end like this, 5 prime to 3 prime strand, without any problem, without any lag or hurdle or discontinuity? Yes. So on this particular strand, because you are continuous DNA synthesis is occurring, right? Continuous DNA synthesis is occurring. Okay, that's why you will call this as leading strand. But on the other strand, can you do same thing, 5 prime to 3 prime? No, right? Then how you are going to do this? You are going to make the strand in this direction, right? Primers and all I am not drawing to save time, but later when you will read, you have to read all that, or when we will discuss that in detail in theory, we will discuss that, right? So you can see this strand is discontinuous, right? You can see small primer was formed, then your DNA, then primer, then DNA, right? So this is discontinuous strand. Okay, and because of discontinuous DNA synthesis, what is the name of this strand? You will call this strand as lagging strand. Okay, you will call this strand as lagging strand. Okay, so you understood these two options, just check they are correct only, right? Previous class may we have learned that DNA replication always occurs in semi-conservative method. What is the meaning of semi-conservative method? This strand which you have written template strand, this is already parental strand. This is also parental strand, but this is new. Okay, this is new. Okay, imagine that both will be formed, right? What will happen? What will happen? One strand of DNA will be always old, one will be new. This is called semi-conservative method of DNA replication. All these points are covered in NCERT very beautifully. Okay. Only one point is wrong here, that is eukaryotic genome is replicated within 30 minutes, which is absolutely in vague. We cannot consider this as correct, because if you know that cell cycle and cell division chapter you have learned, it takes 24 hours for a human cell to divide, to complete its cell division, right? If it is taking 24 hours for cell division, 95% of that time is only in interphase, and one of the most important phase of interphase is S phase, during which all this happens, right? So in any case, we cannot consider 38 minutes only it will take for entire genome to get replicated. This is absolutely wrong. The correct value is not provided in NCERT, so you don't have to learn it, right? But the three statements which are given in this question are directly from NCERT, which you should know, and you have to eliminate them as correct and get to the incorrect statement, right? So there is a strategy, there is a method to solve questions. There must be 200 questions for one topic, but that that doesn't mean that every question will have all four statements which are directly coming from NCERT. There must be one or half of that statement which will not be from NCERT, right? But you have to be very much, you can say, sure that what you have learned in NCERT, that is pakka, or that is absolutely correctly fit in your brain, right? Then you can easily eliminate, avoid the traps and wrong statements which you don't have any information for, but because you have correct information of NCERT and little bit extended information, okay, you can solve all questions. So I hope this is clear to you. I will bring some more questions in the next class. Thank you, keep studying. Okay, bye.